Good morning, good afternoon, or good evening, everyone, depending on where you're joining us from around the world. My name is Esteban Romero, and I lead the webinar programs at ShareVault. It's my pleasure to welcome you to another one of our many life science webinars. Over the past several years, we've hosted webinars covering licensing, partnering, valuation, and therapeutic topics such as oncology, bispecifics, gene editing, and precision medicine, to name a few. Today, you'll hear about some remarkable advances in immuno-oncology, and you'll get a taste for what's to come. On behalf of the many contributors who have collaborated to make this event possible, I thank you for joining us. This is the 81st in ShareVault's webinar series, and today's topic is What's Hot and What's Not in IO 2021. Let's begin with our partners. Cello Health Bioconsulting is a knowledge-based consultancy deeply rooted in science and the unique and ever-evolving challenges and opportunities facing the biopharma industry. Cello Health Bioconsulting is known for unconventional insight, forward-thinking, independent, objective, and actionable strategic advice to realize value across all therapeutic areas and stages of development. To learn more, please visit their website at cellohealthbioconsulting.com. Cello Health Bioconsulting will host the 32nd Annual Cancer Progress Conference virtual on May 4th through the 6th. Cancer Progress now offers partnering to enable meaningful connections with oncology innovators, developers, and investors. Cancer Progress provides an unsurpassed oncology learning and networking experience based on the quality of information, coverage of issues, and value of professional contacts. Thanks to the generosity of their sponsors, Cello Health Bioconsulting is providing complimentary registration. Be sure to visit cancerprogressbycellohealth.com to learn more and to register. Today's webinar is hosted by ShareVault. Many of you in the audience may already know about ShareVault's leadership in the life science industry as a provider of secure, cloud-based business file sharing software and virtual data rooms. ShareVault is commonly used in a variety of due diligence applications for managing regulatory submissions and completed clinical trial master files. What you may not be aware of is that ShareVault is now being used for securely monitoring clinical trials, especially with the recent travel restrictions. Additionally, ShareVault also complements the tools you may already be using, such as Box, Dropbox, Google Drive, Office 365, SharePoint, and DocuSign. ShareVault customers experience a positive business impact because we accelerate due diligence, reduce risk, increase productivity and efficiency, and we reduce cost. If you haven't experienced our cloud-based solutions firsthand, please reach out to us to schedule a demo or set up a free trial. Visit us online at sharevault.com. It is my pleasure to introduce you to our moderator, Linda Pullen. Linda is the founder of Pullen Consulting, which helps in all aspects of business development, from fundraising and partnering. Linda and her team have an extensive deal sheet from company acquisitions to phase 3 to preclinical candidates to technologies. Linda started in the industry in drug discovery, and she led teams that put a multi-billion-dollar antipsychotic and other molecules into development. In business development, she's been on the large pharma side at AstraZeneca and Amgen, and on the smaller side at Kozan Biosciences. Now, for over 14 years, Linda and her team have served as business development consultants to a wide array of biotechnology and pharma companies. The team at Pullen Consulting writes a free monthly newsletter, Pullen's Pieces. Sign up at pullenconsulting.com. You'll also find Linda's work on our resources page. Linda, thank you for proposing and developing today's topic and for leading the upcoming discussion. The podium is yours.
Thank you, Esteban. Welcome, everybody. I'm delighted to be here, and I'll kick it off with asking my co-organizer, the wonderful Jeff Bachman, to say a few words about himself, and we'll continue around the team. Jeff?
Thank you, Linda. It's great to be doing this again. What is this, our 10th or more over the past 5 years? I'm not sure. A lot. Take it on the road soon. Anyway, so yes, I lead the oncology practice at Cello Health Bioconsulting, previously defined health. Oncology is our largest practice area, and I think that's enough said for today.
Thanks, you. Linda. Yes, speaker.
Hi, good morning and good afternoon, everyone. A pleasure to be here. So my name is Astika Gunodhan. I'm a senior biotech analyst at Truist Securities. I cover what I'd like to define as cool stuff in oncology. So I look at immuno-oncology, cell therapy, antibody tech, etc. That's kind of my area. Pleasure to be here.
Thank you. Jake?
So I'm Jake. I'm the CEO and co-founder of Strand Therapeutics. We are a messenger RNA therapy company focused at the intersection of synthetic biology, RNA, and immuno-oncology. Our first drugs are in the solid tumor space, and the mRNA technology is pretty widely applicable into cell therapy and other sorts of areas where I think mRNA will have a big impact in the coming years.
Thank you. Andrew.
Hi, I'm Andrew Pierce. I'm the Vice President of Translational Biology at Crescendo Biologics. We are a company with a primarily oncology focus. We do multi-specific targeting to activate the immune system for fighting cancer purposes.
And Paul.
Thanks, Linda. So I'm Paul Front. I'm the Chief Medical Officer at ImCheck Therapeutics. We are a startup biotech located in Marseille, France. We are focused in the immuno-oncology space, but also have aspirations into the autoimmune space through the targeting of gamma-9 delta 2 T cells and other immune cells that can be modified through the butyrophilin targets of checkpoint molecules. So happy to be here.
Thank you. I think we're going to have a lot of fun here. So let's go to the first slide. Here you can see the top selling cancer drugs a little more than 10 years ago, none of which were IO. In 2020, we see the magnificent performance of Keytruda and Opdivo, really bigger sales than any drug in 2010, and they're forecast to continue to grow. So that kicks off the IO, and the growth of these inhibitors has really been because they are such wonderful backbones for combination. Are there any other IO mechanisms visible on the horizon that can be such a backbone? Jeff, you want to kick that off?
Oh my, I'm sure we would all like to hope so. And unfortunately, it's not clear that in the immediate horizon, we're going to see anything, I think, that will be a true backbone if we really define it as being used everywhere, right? I mean, I think that terminology really derives out of the old origins of kind of current modern oncology, right? With taxanes, for example, as being backbone, pretty much part of every doublet or chemo regimen. So, it'd be hard to see something displacing. Maybe some new agents might have a chance to come in and play alongside of checkpoints in the nearer term. TIGIT is probably, most on everyone's kind of radar. It's got breakthrough. It's a Roche Genentech drug, so it's going to be positioned with the Tizo. We'll see how that kind of helps the Tizo play against Keytruda in particular, but also Opdivo. I wish I could be much more optimistic. I tend to be a bit more Pollyanna because it's hard to lose that kind of science geekiness. Kind of as Astika said, lots of cool stuff. But when you look at the history of the past several years, IDO bust. Not to say it's completely gone and out of the picture. We just had the news today on IPOs from GSK. Obviously, the Merck KGA and GSK partnership around the TGF beta trap, PD-L1, still active, but of course, big stumble in a big kind of head-to-head trial in lung. And only a few weeks ago, the TLR9 from Adera had another hit. Had a couple stumbles over the past several years. So, the litany of kind of IO combinations that are not working, I think is frustrating to all. There's lots of people pontificating about why that might be, rushing too much in, throwing speed against the wall, insufficient translational work, etc. But that's not to say there's still not lots of interesting things, particularly early things that are trying to mine that TME, whether that's on the innate, adaptive, or at the intersection of those things. So there's still active programs, with TLR9s, with kind of next-gen STING and CGAS, RIG-I, obviously. And then there's, lots of cool things going on with other co-stimulatory and inhibitory agonists and antagonists, and big deals still being done, like Sanofi with BIOND with their ILT2 or LILRB agent. So, hopefully I'll have a chance to maybe mention, some of these others in the course of the conversation, some other interesting companies that are kind of up and coming in the space, aside from what we have with our panelists. But, but it is frustrating, right? We're talking about small molecules with IDO. We're talking about kind of classic type of antibody stumbling. We're talking about, the first clinical non-cell redirecting bifunctional stumbling in terms of the TGF-beta trap PD-L1. So, that's frustrating, I think, for the field. So, I think everyone is putting a lot of hopes onto TIGIT, that's for sure. So, but I'm sure many of the other panelists would like to add into that.
Folks want to give a more optimistic or add to Jeff's pessimistic view?
I can add something here. Jeff, you covered a lot of very, you covered some very good points here. But I was also thinking Bristol has Bristol has signaled that we're going to see some positive LAG-3 combination data. So I'm looking forward to that. I think that there's a bit of a silver lining there. They've turned that program around. And one of the other ones I think that maybe is underappreciated, but it's starting to show some really cool translational work, is CD40. We saw some of the data that was presented just at AACR. We've been following also what Apexigen has done in their recent publication. From a mechanistic standpoint, I look at that and say, wow, that's doing some really cool stuff. Is that ready too soon to call it the backbone? Yes, probably. But at least there's some really cool things happening there. And in the least, we know we'll get some sort of a positive trial readout from the LAG-3 this year.
Yeah, well, thanks. I didn't want to cover the whole universe, otherwise no one else would have things to say. But thank you for that. And I did try to answer very much focused on kind of Linda's question about true backbone. I certainly am hopeful that there will be other additional agents to expand in various combinations in various settings. I would really like Apexigen to work because we worked on that program for them when it was early, many years ago. So would love to see that work. Of course, pancreatic is, one of the toughest places to play.
That's great. Okay, let's go to the next slide, which talks about the VC dollars going into Series A, dollars and numbers of deals. And as you can see, VCs have been investing a lot into early opportunities. Presumably, a lot of these are new approaches. One of the things I saw a little while ago now, it was the cancer vaccine in GBM making some buzz. Is there a paradigm shift with vaccines? Is there any new IO approach or theory that is a paradigm shift? Estika, you want to start that off, please?
Yeah, Linda, I'm glad you mentioned cancer vaccines because when I speak to investors and we try to discuss cancer vaccines, people kind of have a bit of a visceral reaction to that, and I think it's also just a case of there's been just disappointments, but as you highlighted, that recent phase 2 data looked really interesting. I think there's other companies that are starting to produce some initial data now. Another company that I cover, HOOKIPA, for example, some very promising hints there and showing actual the kind of immune stimulatory activity, upregulating and increase in CD8 T cells was seen in some of the patients' data that they produce. I think the cancer vaccine space is due for that revival. That's it. That's been on the investor side, on the KOL side and on the academic side. That's always been a hot area. It never fell out of favor. People have just always been very excited about the actual potential that cancer vaccines offers. So there's a bit of a discrepancy there between those two bodies. But yeah, I'm really psyched about cancer vaccines, and I think maybe this year we'll see something really cool emerging from that, and that building some momentum.
Wonderful. Anybody else want to weigh in on cancer vaccines?
I'll wait to give other people a chance. One of my favorite topics.
I can. I'm not an expert in the vaccine field, but I think what we've seen with the development of COVID vaccines really heralds that that these have arrived from a technical point of view. And I think if I know the numbers correctly, the previous record for vaccine development was 4 years for mumps, and now we've just seen an explosion of approvals in the 1-year time frame. So I think that the platform is ready to go, and it would be lovely if that could be applied successfully in oncology, obviously.
Yes.
Well, I think that's a good point about the important thing when talking about cancer vaccines, and having been talking about them going way back to the mid 2000s, so before the dawn of the modern age of immunotherapy. I've been enamored of them, but also have seen so many kind of failures along the way. But I think you brought up an important point. It is important to make the distinction between what you're targeting, right? So where these antigens are coming from. Are they neoantigen, which is the latest flavor, or a classic cryptic neo-fetal antigen, et cetera? And then what is the immunization platform? So mRNA, very facile. Let's hope that, BioNTech, CureTech, Moderna, others, are able to pull that off. But there's still people working with peptides, people working with even whole cell gamishes still from cell lines, people harnessing them to chaperones like heat biologics. So still lots of different flavors of those immunization platforms. And right now it's just not clear, how important. Well, they clearly are important, but so is the choice of the antigen. So, we'll see where we get to between particularly the predictive algorithms and how well they work with the companies still doing that versus more functional approaches like Genosha and other places.
And what about the willingness of people like Merck and BMS to do combinations, given the relatively small effects that most of these vaccines have shown as single agents?
But so I'll actually jump in a bit on still on vaccines, right? So there's a couple different ways that people talk about what a cancer vaccine is, and hell, I've even had like our cytokine-based platform called a cancer vaccine, because in the end of the day, it's creating a prime T cell response, and people are some investors like to call that a cancer vaccine, and then have this same argument about vaccines. So there's kind of like broad antigen, right? Sort of some of the stuff I think even BioNTech is doing in leukemia, where they're taking like broad common antigens in cancer and trying to immunize against that. I think in that case you have the issue of is that antigen really as broadly mutated as you think it is? And then you have what Jeff touched on, which was neoantigen, and some of the work that BioNTech and has done in Gritstone, I think, as well, where they're trying to do personalized vaccines, which I think is completely getting us away from what I think the goal of medicine and getting IO therapies cheaper should be, right? I mean, if every single tumor we have to take out or biopsy, sequence, and then make kind of a de novo multi-epitope neoantigen vaccine against each person's tumor, as like sexy and Star Trek-y as that approach is, it's just completely unscalable. I think, you'll have like maybe 5 centers across the world that can do that for the richest people among us or the most desperate, and everyone else will never be able to reach it. And so I think when we talk about like if we're looking at this slide and we're looking at where new money has been going into oncology, besides the fact that people have completely changed the definition of what a Series A is in the past 10 years, we're now looking at this new. Everyone's looking at how can we become multi-mechanistic? And also, I'd be curious, I don't know the data that was pulled into this, but a lot of companies that are doing quote unquote oncology deals are actually not just oncology companies, right? Like you could probably take our Series A and classify it as an oncology deal because our lead product is in oncology, but our pipeline spans cell therapy, it spans into rare disease. And so I think that the birth of platform therapeutics are going to end up like fundamentally skewing this data. I think the broader scale, and I know this panel is about oncology, so I won't belabor this too much, but the broader scale that we're seeing within the new startups and new startup financing is it's almost not good enough to be to at least be a hot company that's coming forward with technology that's in one single disease vertical, and especially not in oncology, just because there is, as Jeff said, so many different therapeutics that are underlying there. So I think if you come forward with a oncology or cancer vaccine nowadays, then you need to be a company that's like Moderna, that's like BioNTech, that has, a broader scale platform that says, this is a vaccination platform, so we can do cancer vaccines, we can do infectious disease vaccines, we can try to even do autoimmune based immune tolerance sort of vaccine approaches. But I think like just sitting with a single kind of vaccine play where you're going to show up and try to, elicit an immune response against cancer it's not going to be enough. You're definitely not going to show up as a blip on this sort of like Series A oncology chart. There's some legacy companies that are still around, but I think that those are going to fundamentally be combination plays, and the sort of phase 3 results that we've seen when good phase 1 and phase 2 kind of anecdotal data then actually goes into a diligent phase 3 within the vaccination space has just not looked good in oncology. So,
well, and you touch on an interesting thing. I mean, the investments in platforms has waxed and waned over many years and decades, and we certainly are in a period now where it is the re-rising of platform companies, whether that's due to mRNA and the ability of the quick accelerated development of the COVID vaccines or something else. But certainly, I agree, Jake. I think that vaccine that platform companies with the diversity of being able to deploy that. And of course, there's lots of diversity inherently in oncology, let alone going into infectious disease or AID.
So Jake, on this graph, you are absolutely right. This represents companies that include oncology, but may not be only oncology. So that is correct. We also got
the other part is like the changing definition of a Series A, right? Flagship will launch a company, plow $40 million into that company in complete secrecy, and then bring in other investors for $150 million round and call that a Series A because it's the first time everyone's hearing about the company. I think in 2010, that wasn't happening, right? And so when you look at total Series A dollars being up 8x, I mean, if a company is going to call a $50 million round a seed round, and then the next raise a Series A, it's really just kind of like it's just semantics, right? To what people are deciding to call their Series A. So people are calling $4 million rounds pre-seeds now, $20 million rounds seed rounds. We did a $6 million seed round 4 years ago, and people told me that I was being ridiculous by not calling that a Series A. Nowadays, that's like a pre-seed apparently. So I don't know.
Yes, definitions are pretty messy. I agree. We had a question from the audience about if the mRNA approach would make personalized cancer vaccines affordable. Go ahead.
As the mRNA shiller on this panel, I will be the first person to respond to that, I think. So I think mRNA vaccine approach definitely makes it more attainable, right? And that's why Gritstone is using an mRNA approach. That's why BioNTech has been, I think very successful in this sort of turnaround. But again, like just logistically speaking, it can't be where we think the end of oncology treatments should be. It's not where I would want to, in a perfect universe, if you could control like what the future of oncology is going to look like, obviously we need cures. And if personalized vaccines are the end all be all and just blow everything else out of the water, then let's figure out how to make those turnarounds happen. But it's not really like a great future prospect. For example, BioNTech's cancer vaccine program that they have, their personalized cancer vaccine, personalized neoantigen program, right, is like a 3-week turnaround. Biopsy the tumor, you run the mutations through some computational algorithm. It picks out a couple neoantigens that then goes into a vaccine design that then gets made kind of de novo, that is then shipped back and put into the patients in kind of a new setting that these patients have, this drug has never in this combination been used, right? It's to this patient's particular mutations. I think that's very futuristic. I think it's very amazing. I think it would be basically impossible to do that approach with any other modality besides messenger RNA, but I still think it's messy. And I think that it doesn't make it more affordable because, what's affordable about the mRNA vaccines for COVID is that we're making billions at a time, right? And batching in manufacturing, especially with messenger RNA, because it's a wildly scalable, cell-free synthesis reaction, is always going to be able to drop that cost. But if we're talking about making single batches for individual patients, yes, there's manufacturing innovation that can happen. All of the cool things that we're seeing go into the cell therapy world on the manufacturing side now, we could do for messenger RNA. There's companies like Nutcracker that are promising to do, microfluidic assembly of single, mRNA doses at the point of care. All of that may be a future. I don't, it's not where I'm placing my bets in my personal investing either, but it is, a possible future. I just think that there are a lot of other options. And of course, now I'm going to be incredibly biased because my company is developing off-the-shelf mRNA therapeutics for solid tumors. So, take everything I say with a grain of salt and self-interest. But the reason that we're doing that, because I had full control over which direction we initially went in, is that I believe that immunotherapeutics and being able to achieve T-cell responses against tumors will be much more powerful if we can create off-the-shelf modalities that can just kind of build on what we already know about IO responses. And what that allows us to also do is, even in the best case scenario of BioNTech or Gritstone's like neoantigen approach, they have to take thousands of mutations that go into a cancer and boil it down to the 5 best, most immunogenic antigens that can happen. But if you can go into a tumor and create an actual T-cell infiltrating response, combine that with a checkpoint inhibitor, kind of blow up the tumor cells and flood the tumor microenvironment with neoantigens, that opens up a case where you have all of those neoantigens, all those thousands primed out into the microenvironment where the T-cells can now pick up any number of those different neoantigens and actually decide for itself or really just take a bunch of those neoantigens up and form these stronger immune responses. And I think that's just in the end, both going to be a more effective therapeutic strategy, but it's also just going to be more scalable, right? We could make a single or a couple or, a cocktail of different messenger RNA drugs that all work for patients through an immunostimulatory approach, combine that with checkpoint inhibitors and roll that out into clinics across the world, doing batch construction of all of these RNAs, which will drop at least the manufacturing cost. And then we're more in the realm of like traditional therapeutics pricing and figuring out how do we increase access to these drugs to people who need it the most as well.
A couple of comments. One, I'm gonna rib you, Jake, because what you just described sounds like in situ vaccination. So
exactly. That's what I'm saying. I said that, right?
Yep. So, but I do want to pick up on what you said, which I absolutely agree. And the question that was asked is, I think what's important to make the distinction between cost of goods and what you could do to do good in the world versus what historically has been done and likely will continue to be done for some time, particularly in the US, until such time as one is forced to do otherwise. And that is pricing at oncology prices, pricing at parity pricing. So, predicate oncology pricing, even if you can manufacture something with a phenomenal cost of goods, historically, you have not seen anyone coming out with a appropriately priced product that's priced on that price of goods, right? Those products get priced by the oncology pricing parity. So, not to turn this into that big discussion, but I think we just need to be careful there.
I think that's a topic I have strong feelings on, but I think we could get off into all sorts of.
But I think the most important thing is to think about, which is not, instead of focusing on the cost, let's talk about the value, right? Let's talk about getting away from incremental value of adding, not to say that any incremental value is not important for a patient, but from the standpoint of, if you're going to charge $150K or, for a biosoluble biologic or $500,000 for, an autologous cell therapy, let's be darn sure we're setting high bars, not just regulatory bars, but high bars for what those have to do for patients.
Well, and Jeff, I won't jump. You know, I love the drug pricing conversation. I won't jump into it, but I think what I was really harping at was the fact that if you look at like viral based and cell therapies, they're not priced in the oncology bracket of $75,000 to $100,000 pricing,
Yeah,
which
absolutely.
is what off the shelf mRNAs could be priced at in the future, right? If they're made at a batch. But if you make a de novo one, then you're looking at each drug going into a patient costing hundreds of thousands of dollars to manufacture. And then you're back into this crazy realm of where the new generation of therapeutics are. So I'm talking about that sort of a pricing, but let's not dive down this pricing
Well, I
rabbit
also
hole.
want to make sure we get Paul and Andrew into this discussion. So Linda, back to you.
Let's go to the next slide real briefly, which talks about partnerships. And again, the trend to both increasing numbers and values by molecular type. And this, I think, is a good intro into cell therapy, because cell therapy is one of these that has really exploded in frequency of deals and frequency of drugs in the pipeline. And Jeff commented on incremental value, and cell therapy has been a model of incremental improvements in the technology, particularly for all the T-cell companies. So how many companies can we have focused on T-cell therapy? And is there a new cell type that will take over from T-cell therapy? I'm going to ask Paul to talk to this one.
Yeah, well, I think the technology advances and the realization of off-the-shelf cell therapy is one of the driving forces behind the interest and kind of resurgence of cell therapy in oncology. And I think, initially with the knowledge around the T cells and the alpha beta T cells, how to make CAR Ts, was driven by academics. And there really wasn't a lot of focus put on to the second generation now. It's really kind of the innate immune system is becoming the cell therapy playground now, with the NK cells. You've got gamma delta T cells, as you mentioned. There's the macrophages are now, charisma's in play with CAR macrophages. Who would ever thought you'd say that together as a therapeutic? But these are some really impressive technological advances, but based on a lot of scientific understanding of what is the potential of each of these cell populations, and how do you get the most out of them with some of these technological, things that you construct on top of them, generate, genetically modified to withstand chemotherapy, like IN8Bio with their gamma delta program, right? You've got the AFAMED data where they've got these cell engagers that are complex to their NK cells. They were a hit at AACR. And again, as Charisma came out, they dosed their first patients in their CAR macrophage program as well. So I think, to me, there's a new era in cell therapy coming. And I think, as you mentioned, gamma deltas now are coming into play. There's two flavors. There's the gamma 9 delta 2s, which are innate, which used to be incisors. For those of you who don't follow the name changes, there's phosphogam. There's a couple of other assets, and gamma deltas are doing the delta 1s. They both have very different profiles, but provide unique distributions. The gamma 9 delta 2s circulate in the blood, so they're activatable to drive into any tumor or hematologic malignancy, while the delta 1s are more tissue resident and kind of have that homing built in so that they reside in some of these barrier organs, so the gut, the skin, the lungs, where they're exposed to the environment. So it could be really targeted into those types of cancers that affect those types of tissues. So it's early days in the gamma delta space, but there's a lot of companies flocking there, and they've got a lot of big deals with big pharma, as you mentioned. There's these deals going on now for Takeda, Novartis, Gilead. They're all in the gamma delta T cell space with these partnerships with some of these companies that are still yet to really get any legitimate clinical readouts yet. But I think the day's coming where we're going to see whether they live up to the potential promise of, again, taking the advantage of these innate immune cells that have a very different profile, and then partnering them with, is there a way to bridge it to the adaptive immune response with checkpoint inhibitor combinations, or the gamma deltas do it on their own because they have cytokines that they secrete. So there's really a lot of mechanistic potential built into the gamma delta T cell space, which I think has driven a lot of the fundraising, a lot of the the money and partnerships that we've seen over the last couple of years.
Yeah, and I'd actually like to amplify on that, but picking up on what Jake brought up, because, you know, on the one side, there are these, and we're actually going to have a panel at Cancer Progress on this, which we call Battle of Modalities, right? Because on the one hand, you have people busily working to go from auto to allo. You have people kind of working at the interface, Arcellx, Zyphos, others who are trying to create these adapter molecules so that you can have an off-the-shelf cell, but then you can fine-tune to any particular target antigen and tumor as needed. That makes it a little more facile, perhaps a little more cost-efficient, to Jake's point. But also, you've got a lot going on with just, you know, kind of classic, whether cell redirecting kind of soluble biologics or multifunctional biologics that aren't necessarily redirecting cells per se, but they are, shall we say, in quotes, reprogramming, right? They are kind of the next generation beyond Keytruda and Pembro. And that's kind of a segue maybe for Andrew. I'd like to hear some of your thoughts about this, because this goes back also to something that Jake said, right? Because all these things do offer the opportunity for getting that proper milieu in the TME on top of a foundational backbone of a checkpoint, and maybe souping that up so that you do get sufficient immunogenic cell death and maybe sufficient kind of activation and de-repression and stimulation, so that just with those soluble biologics, you can get a sufficient and significant improvement in outcome just above and beyond, say, just, you know, TKIs plus checkpoint, or chemo plus checkpoint, or checkpoints alone. So, Andrew?
Yeah, so we certainly hope so, and that's where our efforts are currently focused. In the general case, I think you could sort of group the bispecifics and the ADCs together on this 2020 chart to a certain degree, and it's around focusing the therapy to a specific tumor marker or specific tumor microenvironment. In the event where systemic toxicity precludes systemic dosing in a non-compartmentalized manner. So I think we've had some tantalizing hints from early attempts. For example, our platform is centered around CD137 or 4-1BB agonism. And there's a history with monoclonals like urelumab that are systemically activating, and it looks promising, but you just can't get to the finish line because you have deal-breaking on target but off tumor toxicity in that case around liver, and people are still having to play with that. I think one of the hopes around the bispecifics and the ADCs is to target to the specific tumor and microenvironment and avoid these sort of systemic effects. And to a certain extent, some of the bispecific engager molecules are could be classified in this same manner, where you're looking to restrict the activity of the T cells to a molecularly defined compartment, typically tumor antigens, but it wouldn't necessarily be so if you had something that was tumor supportive, strictly speaking.
Yeah, great.
And
actually,
Go
ahead, Linda.
I think one of the arguments about cell therapy versus bispecifics or ADCs is cell therapy can bring many different things. Are you thinking toward a single biological non-cell thing delivering more than two?
You get into complexities of scale with these sort of things. A lot of the platforms, including ours, are modular, and in theory, you can tie together as many specificities as you could care to imagine. In practice, though, I think there needs to be a well-defined mechanistic rationale, rather than simply here's a bunch of things, let's hit them all at once and hope that it works. Not only for scientific reasons, but even with these modular approaches, there are complexities that scale in the manufacturability of some of these things. So I think you really need, bispecifics, I think that's cool. That's well understood. I think that's getting to the finish line. Trispecifics, less convinced, and beyond that, I think you're sort of into the wishful thinking realm at the moment.
And I would just add that, I mean, there's still plenty. So, this is not to contradict my earlier statement, because that was what about things becoming backbone. But I think there are lots of interesting next gen targets that even, let's call it monospecific, that standard antibodies, or fusion proteins even, will have the potential to add value to, conceivably in conjunction or relative to the use of checkpoints. I mentioned BIOND before. There's also Immunos, who's also working in this space of kind of these LILRB families that affect both T cells and NK cells and Tregs and MDSCs, et cetera. Or even from the standpoint of where I kind of started about this kind of battle of the modalities, there are cell therapies trying to, actually deliver, gamma delta cells. And then, Paul, you did a very soft sell. And then it was like ImCheck, which
right. So right. I mean, the issue with the cell therapies that I think is still a challenge, right, is how do you ensure they get to where they need to get to, right? You're infusing them into the veins. You're sending in a lot of them, but do they have the activation profile? Do they have the address on where they're supposed to be going? And that's one of the things that we've been working on is trying to figure out how do you redirect and activate once they're in the patient, because that's the key to activate them ex vivo. You can change the phenotype, and then when you put them in, they could be in an exhausted state. So it's really kind of figuring out when's the right time to activate them, and how do you do it, and then direct them into the right target tissue, right? And so that's something that we've been focused on is really kind of understanding how to do that once you're in a patient, because that's what has to happen. Because you can throw in as many cells as you want, but if they don't go to the right place, what do you got?
Yeah. And with a very cool target, right? Which you might want to mention, since I think a lot of people don't even know of this cell surface family.
Yeah, well, one of the targets we're working on is a new family of checkpoint molecules, but called the butyrophilins. BTNs is what they're called, and they are expressed on a lot of different types of immune cells, T cells, B cells, macrophages, gamma delta T cells. And with our original program, ICE-TO1, which is now in the IO clinical development, that's exactly what we do. We target the BTN, we activate the gamma 9 delta 2 T cells, they migrate out of the circulation, they show up in the tumor, they have multifunctional activities associated with them, including production of interferon gamma, TNF alpha, heats up the tumor microenvironment, spreads the anti-tumor immune response. So we're really excited about the stuff we're seeing with now CD8s being activated, NKs migrating. So it is this kind of, like I said, this bridge between the innate and adaptive immune response and kind of how checkpoints can be used with other things in the innate world is where I think the bridges need to be made in order to come up with new backbones, one being in the innate world, one being in the adaptive side of the immune system. And then how do you mesh those together to really create this twisted backbone that you have both sides covered, and then maybe something special to target a specific tumor type or help it understand a specific target within tumor types like that. So to me, it's a wide open space right now, but right now I think that's kind of the one of the limitations that the cell therapy world is faced. And so I think once you can figure out how to do that, then the cell therapy is going to become much more effective.
Sorry, I was going to offer maybe a devil's advocate point of view here on this, because Linda, you asked, do we need this extra incremental innovation with cell therapy? And I was thinking about this, and I thought, you kind of need to do the experiment. You need to run the regulatory gauntlet to get that kind of to get the experience there. I mean, if you survey the biotech world and say how many people in the biotech community have contributed to the development of an antibody, you'll get quite a few people putting their hand up and said yes. For cell therapy, that's a much smaller crowd, and doing the experiments, generating the data with the least amount of variables to get that notch on their belt could then create a favorable situation that sets you up to maybe then do the really cool multiplexing, all the other stuff that you have to do. A great example is that we've been using TIL therapy in academic centers for 2 decades. And it took Iovance from running the regulatory gauntlet to realize, we need to really rethink the potency assay issues here, right? So actually doing that and maybe another company having another CD19 or a BCMA, it's not maybe that bad of a thing, because they might be setting themselves up to do something cooler later down the line once they have that in the bag.
I think that's a great point, but I know many of my client companies look over at the amount of money per company, and yet how many of those there are, and how hard it is sometimes to tell one company's special sauce from another company's special, or to evaluate the difference in that special sauce. It feels like huge amounts of money chasing small differences. Is that unfair?
No, I was playing devil's advocate, and I know that is an unpopular opinion to burn the capital to get the experiment done. But you know, I can see a situation where it can be.
Yeah, well, I think it gets complicated because we're kind of parsing here, right? Because it's one thing to say, you know, you've got to have, you know, these stepping stones to get, you know, to where you want to get to, and that's not to say that those stepping stones, however, can't have reasonable benefits. I mean, I think we have to define there's incremental and there's incremental, right? There's good incremental and bad incremental, as the pirates would say, right? There's truly things. So I think it's more a matter when that discussion came up of saying, you know, not that you kind of completely going to cure the cancer with what you're doing, but, you know, are you aiming sufficiently high for what you hope to get out for the patients, even with your generation 1 or 2 or 3 that you're doing, whether it's a cell therapy or something else. Granted, cell therapies are much more complex in many ways. So,
but I think one of the things that you mentioned is an important one, right? So are we talking about statistically incremental value, or is this clinically incremental value, right? And those aren't always the same, right? You know, p-value that gives you a positive readout may not mean much to a patient who gets, you know, 2 weeks extra to live, right? So it really, there's a difference. And I think we as an industry need to really focus on what is clinically incremental and meaningful for the patients at the end of the day.
Yeah. And certainly the agency's helping too, you know, especially some of the things around cell therapy where those kind of family of, you know, as they get, you know, in various modifications, you know, you're not starting at square one again, right? So.
Right. Yeah. Right. Let's go to the next slide. And here, one of the big messages is the increase in discovery deals over the last 10 years to JustPoint's platforms. And I think I'd like to ask Jake to talk about the piece of this that is new targets, undruggable targets, and you know, KRAS is our poster child. And Jake, how much of a difference do you think new targets, previously undruggable targets, are going to make in terms of efficacy?
So I think you have two different ways to think about it, right? You have new targets and you have new technologies in order to get at the targets. You have companies like Arrakis and others that are now able to target things at the RNA level, and they can both target proteins at their RNA level that were previously undruggable, and then they can also just target completely new areas, things like long non-coding RNAs or other sorts of RNA structures that no one's been able to even interrogate before. I think that technology is going to actually revolutionize the way that we reattack some of the targets that we already knew were there. In much the same way, I think that new technologies on the gene delivery side, as specifically mRNA here, I'm hold my mRNA sign back up, but I think we're all very interested in mRNA these days. I think messenger RNA also opens the door to what can be done in oncology as well, right? In oncology, you've had a number of successes from the point of, does this protein biologic, you know, does this protein actually have an anti-tumor effect? IL-12 is incredibly effective at getting rid of the tumor. It was just also incredibly effective at killing the entire patient themselves. And so you look at, okay, well, with a protein biologic, you have all of these different issues, right? You have systemic toxicity issues, you have leakage, you have dosing issues, you have everything kind of under the sun that happens when you just start injecting proteins into patients. But now you can kind of reimagine it, right? With new gene delivery technologies or gene expression technologies, you can go into different cell types, whether those are T cells or NK cells or the tumor themselves, and start producing these different cytokines. Ones we already know have an anti-cancer effect, right? And so we talked a little bit earlier, the last slide, a few people were talking about like, well, what is the right amount of money to invest into these different platforms, and they all seem like they're kind of the same. I think what becomes interesting about the new platforms is we get to think about things with a different sort of risk tolerance, right? With things like new targets, like IDO was, right? We're investing a lot of money on scientific risk, on clinical risk, and that has a historically poor percentage of us being able to translate laboratory science into the clinic. Now we have the opportunity to turn that thinking into a different mindset, into what is almost an engineering or an execution risk, right? We know that if we can express IL-12 at a low amount that is not toxic from the tumor, we can create an anti-tumor response. We know that the body will respond that way to IL-12 in the tumor microenvironment. And so now it is, can we achieve that from an engineering perspective, from a biological engineering perspective? And I think that will open the door to, new targets is less interesting.
Is it only Jake's frozen?
Only Jake is frozen, but maybe I'll take up the gauntlet because I think you raised a couple of interesting points that are one person asked two different types of questions, Linda, and maybe I'll talk to them because Jake kind of set the stage, because certainly some of these new technologies, whether they're, like Strand or their whole range of companies that are kind of, some of them broadly grouped in this idea of endogenous reprogramming, title, Spotlight, Aventis, Sana, title just, had the Sanofi deal. Sana had a huge raise and IPO, but the idea of being able to go in, and I think this kind of also where Jake was going, this idea of having this level of engineering where you get multifunctionality. So it's kind of going even beyond where kind of soluble multifunctional proteins are, and engineering in multifunctionality, perhaps controllability, whether that's endogenously controlled or exogenously controlled, turning things on and off. Because one of the questions was asking, well, what about the role of RNAi? Well, certainly you can deliver and knock things down. You can also deliver RNA or other things to have things synthesized locally or in the cell type you want, whether it's IL-12 or 15 or frankly even IL-2. So there are lots of companies that are kind of pursuing this multifunctionality in many different ways with engineering that kind of straddles the soluble multifunctional biologics and the kind of where kind of cell therapy is, but are kind of doing it at this interface within the patient themselves.
I'd love to have you comment on how you see truly new targets and how you worry about validation. We get excited about the new and yet the technical risk. Do you want to give us some flavor on your thinking on that?
Oh boy, Linda, if you're going to ask me about how I worry about validation, we're going to be stuck here for a very long time. But I think the field, the investment community tries to do as best as it can by looking at the preclinical data and the right kind of experiments and the right kind of signals. I don't have a whole lot more. That's a very general statement, I know, but it just comes down to that. We do as much as we can to dig into it, look at the preclinical data. If it's doing what it's supposed to be doing, I think there's an acceptable level of risk that you'll take. Okay, so we can, if it's, let's say, upregulating CD8, great, that's what we wanted to do. Now let's take it in clinic. Now if it fails there, now you have to then ask the question of what went wrong. And that's again where the great translational data will come in and help us out and figure out what to do next. What's not helpful is when companies don't do that kind of translational data and don't generate that kind of stuff that we can actually use to kind of parse out and ask the question of what went wrong here. That's the unfortunate situation.
Right.
Yeah, as a translational biology person, I completely agree, actually, and that it's the ability to take what are perforce due to patient numbers in early clinical phase studies being small, taking what are essentially empirical anecdotal observations and bringing those back to the lab to put a mechanistic scientific framework to understand those. And I think that's really where when things go wrong or go sideways, you have an opportunity to learn and improve, not just for your business, but for the broader community. I often tell my colleagues that, in the event that the drug is a raging success and we go in and we cure people, there's no need for translational biology, right? We'll just all go to the victory party. But in the real world, the success rate for new oncology drugs entering the clinic optimistically would be 10%. So there's massive learnings to be had from the failures. And just as you say, this is it's really imperative that anyone going into the clinic takes full advantage of that.
Yes, thank you. Anything else on that one, or do we move on? The next one is yours, Andrew. I was asking about patient selection, and given that IO candidates do not generally have a simple biomarker, may have some sort of immunoprofiling, but it's a pretty complex thing. How are we thinking about a path toward biomarkers and in IO?
Yeah,
another little question. Yeah.
Yeah, it's a fantastic question, obviously, and I think even from things like you can look at how we've done with the most promising markers historically in IO, which would be things like PD-L1 and now tumor mutational burden, and they're not as straightforward as, here's my gene amplified HER2, therefore I have my HER2 targeting drug and it's all good. There's a plasticity of these markers often where markers for immune activation and resistance can come and can go. I think it remains to be seen how much advantage you can get by taking, for example, contemporaneous patient sampling to inform biomarker status versus the archival FFPE block that happened at diagnosis 3, 4, happily many years ago, now that we've made such progress in immunotherapy, but is that still representative? And I think some of the recent, even around a relatively non-plastic marker like tumor mutational burden, you now see emerging data sets that to say, in some tumor types, yes, this is a marker for immunotherapy, but in other tumor types, it doesn't appear to be so. And I think we will learn from whether that holds up or not, but then understanding why those differences occur, I think will also be very helpful. It isn't obvious to me, for example, why tumor mutational burden wouldn't simply be a universal marker for immunotherapy. It increases the likelihood of neoantigens, and it's sort of going where you'd like to be mechanistically, but I think the biology is proving more complicated. So yeah, tough question, Linda.
Yeah, I know. Some of these graphs that do attempt to look at various biomarkers or profiles, the patients, some are going up and some are going down, and the variability is just astronomical, right? So how do we tackle that? It feels like we really need a lot more grouping of patients, subgrouping. Is that appropriate?
Could be. It's starting to happen now with, for example, next generation sequencing, where you're seeing sort of the first approvals for gene panels, right? A patient who has a mutation in any of this small list of genes. But above and beyond that, I think it is a regulatory minefield that's relatively unmapped, and I don't know. And I think for something like multi-specific biomarkers to predict patient response, someone's got to blaze that regulatory trail. But I don't know who's got the brave enough money to give it a go. You'd have to be a real believer in that. And, so it sort of gets back to our discussion about incremental versus paradigm shifting value changes. And I think it's hard enough to do the incremental advances, if I'm honest. So I don't know that we're going to see, for example, here's a gene signature, treat this person with this immunotherapy molecule. And then it's also fraught, obviously, with the more variables you have, now you have this false discovery problem. And it's very easy to go into a small clinical study, non-randomized, and come up with something that looks absolutely brilliant retrospectively that then completely falls apart when you try to apply it at the next step. And of course, there's exponential costs associated with the next step. So it's tricky stuff.
Yeah. Estik, I want to ask you another hard question. As an investor, how are we investing in biomarkers, diagnostics, and the reimbursement challenge that those things have?
That's a tough one, because I think people understand and are supportive of the use of these kind of diagnostics, because you're just enriching for the right patient population. I think there's a lot of willingness to do that. Where you'll probably get maybe the pushback is, what is the right biomarker to look at? What is the right thing to look at? Because when even with PD-L1, it took at least 100 patients worth of data to really understand that this PD-L1 is it indicative or not. And when we first started seeing the data, we actually saw some companies flip-flop between saying yes, it is supportive, yes, or no, it's not supportive. So in theory, I think the community is behind it. We need to really see the biological rationale, and to get to that point, you need to get a good amount of patients worth of data before we can actually do something with it.
Yeah, most of the noise you hear is, not so much around, I mean, you hear a lot of academic discussion, clinical discussion around, finding those markers or signatures for kind of IO responsiveness, either before the fact or monitoring patients under therapy. But when you look at kind of where the noise is around the money, it's more things like GRAIL and Thrive and things like that, to kind of early diagnosis. And I don't know if that's because of, obviously that's very important, but I don't know if it's because of the technical challenges, many of which have just been laid out around kind of the IO space, or, there's something else behind that. But we certainly need it. No doubt about that.
And I'll add to that, the complexity also is because it's no longer something binary, right? I mean, with EGFR, with HER2, it's pretty binary. Are you HER2 positive or not? Are you EGFR positive or not? Here, that's not the case. There's very different shades as well, and then even if you're, let's say, PD-L1 positive, there's a chance that you're not enough, and maybe that in that 1 to 49% or so of patient group, it's not as predictive. So lots of complexity.
Well, that raises a great point too, Astika, because there are situations, even in those you mentioned, where there is disconnect between IHC and FISH and other things, and what is really HER2 low mean, or is there such a thing as HER2 null? So
one of the things that gets me kind of excited about this actually is some of the emerging approaches to use, for example, PET imaging to measure these predictive biomarkers. And, because when we say this patient is PD-L1 positive, well, usually what that means is this 2 millimeter core needle biopsy taken on this day from this site was PD-L1 positive. And, maybe that's representative of bulk disease, and maybe it's not. And now I think with some of the emerging imaging modalities, we'll be able to have a much better understanding of the systemic nature of the marker status for patients. Some of that is coming forward actually in prostate cancer, for example, around this the new Lupisma radionuclide therapy, which people are pounding hard to get a PET imaging prospective selection marker for that.
Good
And
point.
I could see that being sort of the way of the future for single markers.
There are a couple of people looking at T-cell directed imaging, peptides or other things. So yeah, good point.
Very interesting, yes. Okay. Jeff, I had you down to talk about manufacturing automation versus jumping to allogeneic therapies.
All right. Well, fortunately, Jake laid some of that foundation for me already, but well, I'm certainly no manufacturing person, so that's a major caveat. What I can talk to is more about kind of the science and the engineering that's trying to address those issues, right? Because Not to say that there is not a place and a value for autologous cell therapy and situations where that may be viable. But increasingly, the money, the investments, the deals is focused around trying to, at least for cell therapies, get beyond that, right? What are the two big challenges for cell therapy? It's solid tumors and off the shelf, however you define off the shelf, right? And then we already mentioned that there are adapter approaches, and there's, and then there are more specific, single-use allogeneic, there's using iPSCs. So there's all sorts of things going on to try and facilitate that, as well as different ways of engineering, beyond lenti. And some of this is driven by improvements in efficacy of what you're doing. Some of it is ways to work around not paying stacked royalties for like lenti. All right, so you're using CRISPR or other types of gene editing technologies of which there is a plethora that are being deployed both in rare diseases and also in kind of the oncology world for ex vivo cells. So I think there is a lot of engineering trying to solve that aspect of, and when it does come to the front, and I assume when it does, it'll be multiple things that do. A lot of things will flow from that, right? I mean, the speed with which you can deliver to a patient. And in some of these patients, the kinetics of disease are such that you can't wait even the 3 weeks that Jake already alluded to for some of the situations. You also start to address the cost of goods, obviously. And then that's back to our conversation about, how closely actual pricing, tracks with cost of goods and tracks with value being added, for the patient. So I think we are getting there, because these are just solvable. I always think of, various talks I've heard Dan Chen give where, he often says, these are just, biologic engineering things that can be solved, right? It's not something that's insurmountable. And we will get there. I think at the end of the day, it's just a question of even those, facile, off-the-shelf, live cell therapies, with all their engineering, things that they can deliver, turning things off, delivering. Is that the be and end all? It may be. Many people do think it is, but it's also quite conceivable that some cocktail of classic off-the-shelf things, small molecules, soluble biologics, et cetera, might be competitive doing some of the same things. But again, I also don't want to position it as it's an either or, right? Many of these may be fit to purpose, and there may be situations where your choice is an auto cell therapy, an allo cell therapy, an adapter cell therapy, a redirecting cell therapy, a PD-1 plus some other checkpoint and or co-stim. Right now it's all open to question.
Right. One of the things I thought about recently with a friend is that period of waiting. It's not just the kinetics of the disease, but it's the stress. You're sitting there with a diagnosis, waiting for a treatment. It's horrendous. That is.
Well, it's stress, and it's also cost. I mean, a lot of people have to travel very far to a major center, which also I think Jake alluded to, the degree that you can democratize many of these therapies, that it's not just the major centers and the major cities, but that they can reach out to community centers and hospitals, et cetera, because they are easy to use, more accessible in a payment standpoint, better tolerated, so they have a better kind of therapeutic index and profile, so they're not as scary, right? If you're engineering out CRS and all those other things. I mean, that is the ultimate goal.
Yeah. I want to remind the audience to type in some questions. We'll get to those in just a few minutes. Before we do that, Jake, you want to talk about big data and AI?
Sure. Yeah. I'm not a machine learning scientist, though. We do have a division at Strand now that is working on this into how it influences drug development, right? And so there's a lot of different ways that I think that machine learning and we talk about machine learning in science, it's with the exception of people using it to design actual small molecules, most of the machine learning stuff is really just deep learning, right? It's really just training on large data sets and trying to find like correlations that, you know, no human would have the time or the memory to be able to figure out, you know, reading millions of different things and looking for tiny correlations. But I think that, you know, AI is likely going to innovate a lot in our trial design in the future over the next like 10 years. You know, we talked about biomarkers earlier, just the last slide. And I think that the likely future of, you know, sort of more so than just like PET image or PET imaging in order to find like different distributions of single targets. I think what we'll likely start pushing ourselves towards is how does a genotype map to a phenotype, and then just doing full genotype mapping and full phenotype mapping from those genotypes of individual tumors, because I think that, you know, 1 or 2 or even 3 biomarkers in a tumor is just not enough to really speak to the overarching diversity. I think there's a lot of changes that happen that lead to those phenotypes, different regulatory changes that we as humans just don't really have the capacity to fully understand. And so what I would like to really see is people starting to dig into tumors, start to dig into that heterogeneity, start to dig into understanding of, you know, what is it that causes PD-L1 overexpression on the surface? And if I perturb that in some way, you know, how will that be affected the overarching regulatory network inside? So I think that's one area that deep learning and sort of, I don't know, big data, if you want to call it that, but where it's going to innovate in sort of like how we're able to actually stratify patients, and likely even further, how we're able to design clinical trials. On the drug design front, you obviously have a lot of companies using it to design different sorts of small molecules. Recursion Pharma is about to do an oversized IPO right now. They're one of the leaders in the space. Another one out in San Francisco named Atomwise is another leader. I can't really speak to like whether or not they will truly innovate on the drug discovery process, right? The thesis there is that AI will be able to better predict small molecules that have the structure to bind into certain binding pockets and targeting domains of proteins. I think that they'll have at least, if not better of a chance than sort of the human medical chemistry sort of brute force method we've been using for the past 50 years. Whether or not that will gain outsized returns like people think, I think is still up for debate, right? Will the machine learning design of those molecules in oncology, right? We talked about new targets. Will that actually lead to more efficient therapeutics that can move through the clinic faster or move through preclinical development faster, or have a higher likelihood of being approved, which then lowers the overall cost or increases the amount of price that we can demand at any one time to fund the development of these drugs? That's the thesis. It's kind of yet to be proven, right? Just the ability to more rapidly discover drugs, I think is missing a lot of where drug development burns a lot of its money, which is in later stage preclinical and then eventually going into clinical trials and being able to motivate money into a drug company. I think finally, the area that I'm the most excited about AI and deep learning in is actually in the gene and cell therapy space. And again, I will acknowledge that I am biased because that's exactly what we're doing at my company as well, using AI, machine learning, and most efficiently deep learning to better understand how sequence of different genetic-based drugs influences their behavior in different cell types and different tissue types across the body. I think from, you have companies like Dino Therapeutics out of George Church's company, or out of George Church's lab, which is essentially a company mill, about as productive as Flagship at this point. But Dino Therapeutics spun out of George Church's lab a few years ago. They're using AI to design better AAVs, so design better capsids, which all is about sequence engineering of the AAVs. In Strand Therapeutics, I think we've said this pretty publicly, is that we're also using, we use a lot of different types of mRNA species, self-replicating messenger RNAs, And we have found that all sorts of sequence perturbations and engineering to those sequences can change the behavior of those RNAs when they're in the body. It can change the types of immune responses that they elicit. And we've been able to anecdotally go in and change and find different species that we can use for drug products. But now can we take all of that anecdotal evidence and actually start to diligently peel it apart with deep learning technologies? I think that in gene and cell therapy is going to be where AI really takes off in drug design, just because the amount of unforeseen data, right? The amount of engineering black boxing that we don't understand when we design a messenger RNA and we pick a 5' UTR and we pick a 3' UTR and we pick the codon optimization scheme for a given protein. All of that we've done so far is been fairly crude. We haven't really thought about how to design these therapeutics in a logical setting, but we know that individual changes, Moderna's published on this, changes to the 5' UTR of their modified RNAs impact the translatability of the mRNA. We don't know why. And so it's kind of like a perfect problem for deep learning to come in and actually look at large pieces of data and try to pull out different sorts of design rules that in the end, we may never fully understand why those design rules are the way they are, but we will be able to build better, more specific and more active, maybe less immunogenic drugs. Longer lasting messenger RNAs is a big focus of tissue-specific AAVs are a big focus of Dino, and really, all of the companies that will kind of fill in between to use it in the same way. There's another Church Lab spin-out new startup called Manifold Bio, same sort of approach, but in designing antibody sequences. I think that sort of design at the genetic and protein interface is where we're just going to see an explosion of growth and probably an explosion of company creation over the next half a decade.
Thank you, Jake. So we have a question from the audience that's right up my sweet spot, and that is sort of what is the next big trend? What is the next market disrupting event? And as our investor representative, Astika, you want to kick that one off?
That's a great question. It's a very difficult one as well, because there's so much cool stuff going on right now, and I really think we're in essentially a renaissance period for biotech, and all these different areas just firing on all cylinders. So it's really hard to pick what is the next big trend. But maybe I'll discuss something that maybe in the next few years that I think could really play out here, and is one of many. I think it's the immune cell engagers. Prior to AACR, the last immune cell engager to produce phase 3 data, I think, was Blincyto. Now we saw something for Immunocore. We have potentially pivotal data packages that are being generated by Roche and Regeneron for the CD20, CD3 bispecific there. And then just take a step back. There's so many new configurations and adaptations of this, relatively basic bispecific antibody or bispecific scaffold that's coming out there. And I think that's a bit of a groundswell of new companies, new data that's been generated. So if I was to pick something right now in the near term, I'd say it would be a trend to improve what we're seeing out of the immune cell engager. But that's not saying that there's not going to be others. Of course, as the things we discussed on this panel, cell therapy, mRNA, vaccines, etc. There's just so much going on, but I'll go with immune cell engager for now.
One of the audience questions is about oncolytic viruses.
Oh, as a lapsed virologist, another area I've longed for to have, significant contribution to this story, and unfortunately, sorry, Linda, as an ex-Amgen person, but so far we only have ImCheck, which, is it's not the greatest thing since sliced bread. So, there are lots of different flavors, people working on lots of different viral families, from simply just, again, different viral family putting in GM-CSF, which has pros and cons, to heavily engineered type things like Cyoxis or Oncorus, et cetera, that, start to look somewhere between a virus and a redirecting bispecific and a cell, because there's just so many kind of things being done to them. At the end of the day, they should work, right? I mean, they have, in many cases, tropism to the tumor because of the alterations that happen inherently along the interferon sensing pathways. They are great at, kind of destroying cells and leading to immunogenic cell death. They have DAMPs and PAMPs, so they kind of straddle kind of innate and adaptive immunity. So in theory, you should be getting all of this plus in situ vaccination, et cetera. And then if you start engineering them to carry in or express or, modify, with their own checkpoint or co-stims or other things, and yet, we're still waiting for, the really definitive data with, any one of these many oncolytic virus companies. And there, it's not to say there haven't been deals, lots of M&A and deals have occurred over the past several years across a variety of different kind of viral families. But again, I'm holding out hope, kind of like cancer vaccines, but, to mix my metaphors, show me the money.
Yeah. So another audience question is, has anybody been able to decrease receptors on T cells that lead to exhaustion?
Interesting question.
I could take a bit of a go at this. Actually, is that there's some focus around mitigating T cell exhaustion through agonism of anti-apoptotic, pro-stimulatory, pro-memory molecules, and of course our company is one of those having a play at this. So I think it's a key question actually, and this may be one of the concerns out of the T cell engager field is how do you keep those T cells hopped up and cooking? And but I'm not aware of approaches to decrease the T cell energy molecules on the surface.
On the cell therapy side, there's lots of gene edits going into attempting to address exhaustion, persistence, etc. But I took the question to mean, some exogenous applied, to the patient, but maybe that I'm misinterpreting that.
Well, Jeff, I was thinking of Maxite, which has the karma, which is minimally manipulated so as to avoid the T cell exhaustion.
Well, that's a whole other good point too about, this trend of minimally manipulated. I mean, there are people who even take PDSCs and do some interesting work with company, because I know the founder well. Alloplex, for one, does some cool things. And then on the flip side, there are some interesting technologies to try to, say, increase expression of target molecules, increase expression of MHC, since downregulation, shedding, splicing, escape mechanisms, classic for CD19 and probably other things as well. So, whether it's companies like biologics or a variety of others that are trying, have drugs that apparently, upregulate expression of the good molecules as opposed to downregulating the bad molecules.
Well, downregulating the bad molecules was another question. Is there a single agent that might decrease expression of checkpoint inhibitor receptors?
I think I've seen some gene editing type of approaches as part of the endogenous reprogramming collection of companies that are kind of, doing this, again, going back to the idea of kind of multifunctionality, multiple engineering and things going on delivered in nanoparticles or other things like that, but I'm not sure about things outside of that off the top of my head.
Right. One last audience question was back to why hasn't OX40 and LAG3 taken off?
Well, Ismika, you should reiterate your LAG3 point.
Yeah, I mean, well, I think LAG3 is about to take off. Bristol Myers is fixing to present some data, I think, at ASCO, where they're going to where they've already signaled that the trial had a positive outcome. So I think, there must have been some work that went into that. Perhaps it might have been to do with the dosing, but it sounds like they've been able to do that. In terms of things like OX40 and maybe other things in the TNF superfamily receptor group, we've learned that engaging the receptors and clustering them is a key requirement to actually produce a more optimal higher order signal. And I think companies have gotten behind that and are engineering their antibodies and their products to do it. Jeff, you mentioned our good friend Dan Chen. I mean, IGM Biosciences is one of them, Genmab, all these others, that they'll look at more sophisticated antibody products that are able to do that kind of clustering. So it's that step of innovation that had to happen, and now we start to see that being implemented into these products.
Yeah, that's a great point about, some of these that are trying to address the trimerism or, that you need. So you mentioned some companies. There's also a German company called Apogenics with their platform, a variety of ones that are trying to more match their platform to the necessary modulation for these, particularly that TNF family.
Yeah, I think that's exactly right, actually. And a lot of the early approaches were with very traditional monoclonal antibodies, where the bivalent format is sort of inherently mismatched to the trimeric receptor biology that these pathways are expecting to see. So I think it's a watch this space kind of place.
Any last watch this space before we close this out?
Well, there's no lack of wonders going on. Just hope they have wonderful outcomes for patients.
Right. Well, then let me start by thanking a truly marvelous panel and really appreciate all the thoughtfulness and knowledge of the field that I think was on display today, and very much thank the audience for joining us, and thank you to ShareVault.
Yeah, thank you, everyone, particularly those who are in less hospitable time zones.
Yes, thank you indeed.
Yep. Thank you very much, Linda, Jeff, Andrew, Astika, Jake, and Paul for sharing your knowledge with us. Thank you for the audience for joining us today. Be on the lookout for an email in the next few days that announces the availability of the recording and slide deck. We sincerely hope that the information you received today will be helpful to you. We wish you good health and a wonderful remainder of your day.
Thanks, everyone. Stay safe.
Thanks. Bye everybody.
Bye-bye.
Thank you.
Thank you.
Bye-bye.
Bye-bye.